The green world breathes without lungs, thinks without brains, and remembers without memory as we know it. A silent intelligence woven through root and soil, older than the first human thought, patient as stone, alive in ways we are only beginning to understand. Beneath your feet, beneath the fallen leaves and the dark humus, beneath the visible surface of the forest floor, there exists a network so vast and intricate that it rivals the neural pathways of the human brain. This is the mycorrhizal network, the wood wide web, a subterranean internet of fungal threads connecting tree to tree, plant to plant. In a conversation conducted entirely in chemical whispers, the threads are called hyphae, each one finer than the finest human hair, and they stretch through the soil in numbers beyond counting, weaving between root tips, wrapping around them in gentle spirals, forming bridges of living tissue. Through these bridges, trees share carbon, nitrogen, phosphorus, and water. A mother tree, ancient and towering, will send sugars to her offspring struggling in the shade below, recognizing her own through chemical signatures we cannot see or smell. The forest is not a collection of individuals competing for light and space. It is a commune, a collective, a single breathing organism dispersed across acres of land, thinking slowly, sharing resources, warning of danger through pulses of chemical information that travel faster than you might imagine through the dark earth. In the redwood forests of California, scientists have discovered that the largest trees act as hubs in this network, connected to hundreds of other trees, young and old, of the same species and different ones. These hub trees, these mother trees, are not merely the tallest or the oldest. They are the most connected, the most generous, the ones who have learned over centuries how to share. When a Douglas fir is attacked by insects, it sends chemical alarm signals through the mycorrhizal network, and neighboring trees, even those of different species, receive the message and begin producing defensive compounds in their leaves and bark before the insects arrive. This is not metaphor. This is measurable, repeatable, documented communication. The trees are speaking to one another in a language of carbon and nitrogen, of sugars and enzymes, a language that predates human language by hundreds of millions of years, and the conversation never stops. Even now, in the darkness, in the cold, in the winter silence, the roots are touching, the fungi are threading, the chemicals are flowing, and the forest is aware. The fungal threads that form this network are themselves a wonder of biological architecture. A single teaspoon of healthy forest soil contains several miles of fungal hyphae, invisible to the naked eye but present in densities that would astonish you if you could see them. These threads are not passive conduits. They are living tissue, growing, branching, dying back, responding to chemical gradients in the soil, seeking out nutrients, seeking out roots, forming partnerships that benefit both fungus and plant. The fungus receives sugars from the plant, produced through photosynthesis, and in return, the fungus provides the plant with water and minerals it could never access on its own. The fungal network extends the reach of the plant's roots by a factor of 10, sometimes 100, allowing the plant to tap into resources far beyond its physical location. This partnership is ancient, older than the first forests, older than the first trees. When plants first colonized the land, more than 400 million years ago, they did so in partnership with fungi. The earliest plant fossils contain fungal structures within their cells, evidence of a relationship that made life on land possible. Without fungi, there would be no forests, no grasslands, no flowering meadows. The green world as we know it exists because of this partnership, this invisible collaboration happening beneath every step you take. The mycorrhizal network is not the only form of plant communication. Above ground, in the air, plants release volatile organic compounds, complex molecules that drift on the breeze, carrying information, When a willow tree is browsed by caterpillars, it releases a specific cocktail of chemicals into the air, and neighboring willows detect these molecules and begin producing tannins and other compounds that make their leaves less palatable, less nutritious, harder to digest. The caterpillars, arriving at the neighboring tree, find the leaves bitter and tough, and they eat less, grow more slowly, die more often. The tree has warned its neighbors, and the neighbors have listened. This happens in real time, within hours, sometimes within minutes. The air in a forest is thick with these messages, a constant exchange of chemical information that we walk through without noticing, a conversation in a language we have no receptors to detect. But the plants detect it. They are listening always to the chemical composition of the air around them, adjusting their metabolism, their growth, their defenses in response to what they smell. Some plants can even distinguish between the saliva of different herbivores. A tobacco plant, when chewed by a caterpillar, produces one set of defensive chemicals. When chewed by a grasshopper, it produces a different set. The plant is not merely reacting to damage, it is identifying the attacker and tailoring its response accordingly. This requires a form of perception, a form of information processing, that we are only beginning to understand. The plant has no brain, no neurons, no central processing unit, yet it perceives, it decides, it acts. The mechanisms are distributed throughout the plant's tissues, in the cells of the leaves, the roots, the stems, in the electrical signals that pulse through the plant's vascular system, in the waves of calcium ions that ripple through the cytoplasm, coordinating responses across distances, across the entire body of the plant. A plant is not a passive organism rooted and helpless. A plant is an active, sensing, responding entity, engaged in a constant negotiation with its environment, with the soil, the air, the light, the creatures that would eat it. The intelligence of plants is not like our intelligence. It is slower, more distributed, more patient. A tree does not think in seconds or minutes. It thinks in seasons, in years, in decades. A tree remembers the droughts of the past by the width of its growth rings, each ring a record of a single year, wide in the wet years, narrow in the dry ones. By reading these rings, we can reconstruct the climate of centuries past, the wet periods and the dry periods, the warm years and the cold ones. The tree carries this information in its body, a living archive of environmental history. And the tree uses this information. A tree that has experienced drought will allocate more resources to root growth, will produce deeper roots, will become more efficient at water uptake, preparing for the next drought which may not come for years or decades. The tree is learning from experience, adjusting its behavior based on past events, which is one definition of intelligence. In the tropical rainforests, where competition for light is fierce, some trees have developed a form of memory that borders on the uncanny. The Mimosa pudica, the sensitive plant, will fold its leaves when touched, a defensive response to herbivores or to the impact of falling debris. But if you drop water on the plant repeatedly, from the same height, in the same rhythm, the plant will eventually stop folding its leaves. It has learned that the stimulus is harmless. This is habituation, a simple form of learning, but it is learning nonetheless, and the plant remembers. If you wait a month and then drop water on the plant again, it will not fold its leaves. The memory persists. The plant has no neurons, no synapses, no hippocampus, yet it remembers. The mechanisms are not understood, but the phenomenon is real, documented, repeatable. The plant is not a machine responding blindly to stimuli. It is an organism capable of learning, of memory, of adjustment. The roots of plants are perhaps their most sensitive organs, more sensitive than we ever imagined. The root tip, the very end of the growing root, is covered in a cap of cells that are constantly being sloughed off and replaced. And within this cap, there are cells that contain starch granules that settle to the bottom of the cell under the influence of gravity. These are the plant's gravity centers, its statocytes, and they allow the root to grow downward, always downward, into the soil, seeking water and nutrients. But the root tip is also sensitive to light, to moisture gradients, to chemical signals, to the presence of obstacles. A root growing through the soil will navigate around rocks, will grow towards sources of water, will avoid toxic chemicals, will grow toward beneficial microbes. The root tip has been called the plant's brain, and while this is an oversimplification, it captures something true. The root tip is a site of integration, of decision-making, of directed growth. The root is not passively pushed through the soil, it is actively exploring, sensing, choosing its path. In a single cubic centimetre of soil, there may be billions of bacteria, millions of fungi, thousands of protozoa, hundreds of nematodes, and dozens of larger organisms like mites and springtails. This is the rhizosphere, the zone of soil immediately surrounding the root, and it is one of the most complex and dynamic ecosystems on Earth. The root exudes sugars, amino acids, organic acids, and other compounds into the soil, and these exudates attract microbes which colonize the root surface and the surrounding soil. Some of these microbes are beneficial, helping the plant acquire nutrients, protecting it from pathogens, stimulating its growth. Others are neutral or harmful. The plant actively shapes the microbial community of the rhizosphere by the compounds it exudes, favoring beneficial microbes, deterring harmful ones. This is chemical communication on a microscopic scale, a conversation between plant and microbe conducted in the language of organic molecules, in the darkness of the soil where no light penetrates, where the only currency is chemistry. The oldest living organisms on Earth are plants. In the White Mountains of California, there are bristlecone pines that have been alive for more than 5,000 years. They were seedlings when the pyramids were being built, saplings when Rome was founded, mature trees when Columbus sailed to the Americas. They have survived droughts, fires, ice storms, insect outbreaks, and the slow grinding of geological time. Their wood is so dense and resinous that it resists decay even after death. Fallen bristlecone pines can lie on the ground for thousands of years, their wood still intact, still hard, still preserving the record of their growth rings. These trees grow slowly, sometimes adding less than a millimetre of width in a year, conserving resources, enduring. They are not the tallest trees, not the most massive, but they are the most patient, the most enduring, the most successful by the measure of longevity. They teach us that survival is not about speed or size. It is about resilience, about the ability to endure, to wait, to persist through conditions that would kill faster-growing, more ambitious organisms. There are seeds that can wait for centuries, for millennia, buried in the soil, dormant, metabolically almost inactive, waiting for the right conditions to germinate. In the permafrost of Siberia, scientists have found seeds of the narrow-leafed campion, a small Arctic flower, that were buried by squirrels more than 30,000 years ago. The seeds were still viable, planted in soil, watered, given light. They germinated and grew into healthy plants that flowered and produced seeds of their own. 30,000 years of waiting, of suspended animation, of patience beyond human comprehension, and then, given the chance, the seed awakens, the embryo stirs, the root emerges, and life continues. This is not mere survival. This is a form of immortality, a way of transcending time, of waiting out ice ages and climate shifts, and the rise and fall of entire ecosystems, waiting for the moment when conditions are right, and then seizing that moment, growing, flowering, reproducing, ensuring that the lineage continues. The Amazon rainforest, often called the lungs of the earth, produces approximately 20% of the world's oxygen. but it also consumes nearly all of that oxygen through respiration, the metabolic processes of the plants, animals, fungi, and microbes that live there. The net contribution of the Amazon to the global oxygen supply is close to zero, but this does not diminish the forest's importance. The Amazon is a vast engine of the water cycle, transpiring billions of gallons of water into the atmosphere every day. Water that forms clouds, that travels on the wind, that falls as rain hundreds or thousands of miles away, sustaining agriculture, filling rivers, shaping climates. The trees are not just producing oxygen, they are moving water, they are creating weather, they are stabilizing the climate of an entire continent. A single large tree can transpire hundreds of gallons of water in a day, pulling it from the soil through the roots, moving it up through the trunk in the xylem vessels, releasing it from the leaves through the stomata, the tiny pores on the leaf surface. This is a hydraulic feat of staggering scale, powered entirely by the evaporation of water from the leaf surface, which creates a tension, a negative pressure that pulls water up from the roots, up through the trunk, up to the canopy, sometimes more than 300 feet above the ground, against the force of gravity without any pump, without any moving parts, using only the physical properties of water and the elegant architecture of the plant's vascular system. The color green, the color of chlorophyll, the color of life on Earth, is the color of the wavelengths of light that plants do not use. Chlorophyll absorbs red light and blue light, using the energy of those photons to drive photosynthesis. But it reflects green light, which is why plants appear green to our eyes. In a sense, the green we see is the light the plant has rejected, the light it does not need. The plant is harvesting the sun, capturing photons, converting light energy into chemical energy, into the bonds of glucose molecules, which are then used to build cellulose, lignin, proteins, all the structures of the plant's body. Photosynthesis is the most important chemical reaction on Earth, the foundation of nearly all food chains, the source of nearly all the oxygen in the atmosphere, the process that transformed the early Earth from a barren, lifeless rock into a planet teeming with life. And it happens silently, constantly, in every green leaf, in every blade of grass, in every algal cell in the ocean, driven by the energy of the sun, which has been shining for 4.5 billion years and will continue to shine for billions more. And as long as it shines, the green world will capture its light, will grow, will breathe, will endure. In the deep shade of the forest floor, where only 1 or 2% of the sunlight that strikes the canopy ever reaches the ground, there are plants that have adapted to survive on almost no light at all. These shade-tolerant species have leaves with higher concentrations of chlorophyll, giving them a darker green color, allowing them to capture more of the limited light available. They grow slowly, conserving energy, waiting for a gap in the canopy, waiting for an old tree to fall, and when that gap appears, when the sunlight suddenly floods the forest floor, the shade-tolerant seedlings respond, growing faster, reaching upward, racing to claim the light before the gap closes, before the surrounding trees grow laterally and seal the opening. This is the lottery of the forest, and most seedlings lose. They wait in the shade for years, sometimes decades, growing a few centimeters a year, and then they die, killed by disease or herbivores, or simply by the accumulated stress of living in near darkness. But a few win the lottery. A few find themselves in a light gap at the right time, and they surge upward, and they become the next generation of the canopy, and the cycle continues. The bark of a tree is not dead tissue, though it may appear so. The outer bark is dead, a protective layer of cork cells, but just beneath it there is a thin layer of living tissue called the phloem, which transports sugars from the leaves to the rest of the tree, and beneath that there is the cambium, a layer of cells only a few cells thick, which divides continuously, producing new xylem to the inside and new phloem to the outside, adding girth to the tree year after year, ring after ring. The cambium is one of the most active tissues in the tree, dividing, growing, responding to hormones, to the seasons, to the availability of water and nutrients. In the spring, when water is abundant, the cambium produces large, thin-walled xylem cells which show up as light-colored wood in the growth ring. In the summer, when water is less abundant, the cambium produces smaller, thicker-walled cells which show up as dark-colored wood. The transition from the light wood of one year to the dark wood of the next creates the visible ring, and by counting the rings, we can determine the age of the tree, and by measuring the width of the rings, we can infer the growing conditions of each year, the wet years and the dry years, the warm years and the cold years, the years of plenty and the years of stress. Some trees can clone themselves, sending up new shoots from their roots, creating groves of genetically identical individuals connected underground. The quaking aspen is famous for this. In Utah, there is a grove of aspens called pando, which is a single organism, a single genetic individual, consisting of more than 40,000 stems, covering more than 100 acres, all connected by a single root system. Pando is estimated to weigh more than 6 million kilograms, making it one of the heaviest organisms on Earth, and it is estimated to be thousands of years old, possibly tens of thousands of years old, though the individual stems live only a century or two. The stems die and are replaced, but the root system endures, and the organism continues, a single life dispersed across a forest, a single genetic individual experiencing the world through thousands of bodies, thousands of sets of leaves, thousands of root tips, all connected, all sharing resources, all part of the same living whole. The silence of the plant world is not the silence of inactivity. It is the silence of a different kind of activity, a different kind of life, one that unfolds on timescales we are not accustomed to perceiving. A tree does not move in the way an animal moves, but it moves nonetheless. It grows toward the light, it extends its roots toward water, it sways in the wind, it opens its stomata in the morning and closes them at night. It flowers in the spring and drops its leaves in the fall. These are movements, slow movements, imperceptible in the moment, but undeniable over time. If you could watch a tree grow in time-lapse, speeding up the process by a factor of 1,000 or 10,000, you would see it writhing, reaching, responding, as active and dynamic as any animal, as engaged with its environment, as alive. The slowness is not a limitation, it is an adaptation, a way of life suited to organisms that are rooted in place, that cannot flee from danger, that must endure whatever comes. And endure they do, for decades, for centuries, for millennia, outlasting empires, outlasting civilizations, outlasting the brief flicker of individual human lives, standing as witnesses to the passage of time, as anchors of continuity in a world of constant change. The green world is not separate from us. We breathe the oxygen it produces, we eat the sugars it synthesizes, we build our homes from its wood, weave our clothes from its fibers, derive our medicines from its compounds. We are embedded in the green world, dependent on it, part of it whether we acknowledge this or not. And the green world, in its silence, in its patience, in its slow and ancient intelligence, continues to grow, to breathe, to communicate, to remember, to endure, a living network spanning the planet. Older than our species, more resilient than our civilizations, a testament to the power of life to adapt, to persist, to thrive in the face of time and change, and the endless turning of the seasons. There are seeds sleeping in the soil beneath your feet that are older than your grandparents, older than the oldest living person, older than nations and languages, and entire civilizations, waiting in a state of suspension so profound that time itself seems to have forgotten them. The sacred lotus, Nelumbo nucifera, produces seeds encased in a hard, impermeable coat that can remain viable for more than 1,000 years. In a dry lake bed in China, Archaeologists unearthed lotus seeds that radiocarbon dating revealed to be approximately 1,300 years old, buried during the Tang dynasty, when poets wrote of moonlight on water and emperors commissioned temples of jade. These seeds, when carefully scarified to allow water penetration and then planted in warm mud, germinated within days. The embryo inside had been waiting, metabolically nearly silent, respiration reduced to levels barely detectable, yet still alive, still capable of recognizing the chemical signals that meant water, warmth, oxygen, the conditions necessary for growth. The seedling that emerged was genetically identical to its parent plant from the 8th century, a living bridge across time, a continuation of a life that had paused but never ended. The mechanisms of seed dormancy are among the most elegant solutions to the problem of survival that evolution has ever produced. A seed is not merely a plant in miniature, it is a capsule of potential, a compressed future, equipped with everything necessary to begin life except the trigger. Inside the seed coat lies the embryo, consisting of the embryonic root, the radicle, and the embryonic shoot, the plumule, along with 1 or 2 cotyledons, the seed leaves, which contain stored nutrients, starches, proteins, oils that will fuel the first days of growth until the seedling can photosynthesize on its own. Surrounding all of this is the seed coat, a protective barrier that can be thin and permeable or thick and armored, depending on the species and the strategy. Some seeds are designed to germinate quickly within days or weeks of being shed. Others are designed to wait, and the waiting can last for years, decades, centuries, even millennia. The longest documented seed viability belongs to the date palm. In the 1960s, archaeologists excavating the fortress of Masada in Israel, the site of a famous siege in 73 CE, discovered a cache of date palm seeds in a clay jar. The seeds were approximately 2,000 years old. For decades they sat in storage, a botanical curiosity, until 2005, when a researcher decided to attempt germination. One seed, nicknamed Methuselah, sprouted. The seedling grew into a healthy tree, and in 2011 it flowered, producing pollen that was used to pollinate a modern female date palm, resulting in viable dates, fruit from a tree whose genetic lineage had been interrupted for 2 millennia. Methuselah is a male tree, and his pollen carries genes from a population of Judean date palms that went extinct centuries ago, a genetic legacy preserved in dormancy, waiting for the chance to continue. Dormancy is not passive. It is an active state, maintained by chemical inhibitors within the seed that prevent germination even when conditions seem favorable. Abscisic acid, a plant hormone, accumulates in the seed during development and inhibits the processes necessary for germination. The seed will not germinate until the abscisic acid is broken down or diluted, which happens through exposure to specific environmental cues, cold stratification, fire, passage through an animal's digestive system, scarification by abrasion. Each species has its own requirements, its own lock that must be opened by the right key. This ensures that the seed germinates only when conditions are truly suitable, not during a brief warm spell in the middle of winter, not during a drought, but when the season is right, when water is available, when the chances of survival are highest. Some seeds require fire to germinate. In the chaparral ecosystems of California and the fynbos of South Africa, many plant species produce seeds with hard, resinous coats that will not crack open unless exposed to the intense heat of a wildfire. The fire clears the landscape of competing vegetation, releases nutrients from the ash into the soil, and creates open space with abundant light. The seeds, buried in the soil, are insulated from the lethal temperatures at the surface, but they experience enough heat to crack their coats, and when the rains come after the fire, they germinate en masse, carpeting the blackened earth with green. This is not destruction. This is renewal, a cycle that is repeated for millions of years, a partnership between fire and seed, between death and life. Other seeds require cold. Many temperate tree species produce seeds that will not germinate until they have experienced a period of cold stratification, weeks or months of temperatures near freezing, which breaks down the chemical inhibitors and primes the embryo for growth. This prevents the seed from germinating in the fall, when it is shed, only to have the seedling killed by winter frost. The seed waits through the winter, experiencing the cold, and then when temperatures rise in the spring and moisture is available, it germinates, and the seedling emerges into a world of lengthening days and warming soil, the optimal time for growth. In the deserts of the American Southwest, there are annual wildflowers whose seeds can wait in the soil for decades, germinating only when rainfall exceeds a specific threshold, usually 2 or 3 inches within a short period. Lesser rains will not trigger germination because the seedling would not have enough moisture to complete its life cycle. The seed is measuring the environment, integrating information about rainfall, temperature, and day length, and making a decision, a biochemical calculation of risk and reward. If the conditions are right, the seed germinates, the plant grows rapidly, flowers within weeks, sets seed, and dies, all before the soil dries out completely. If the conditions are not right, the seed waits, and it can wait for a very long time. The Arctic tundra, a landscape of permafrost and brief summers, is also a repository of ancient seeds. Squirrels and other rodents cache seeds in burrows, and if the burrow is in permafrost, the seeds can remain frozen for thousands of years, preserved in a state of near-perfect suspended animation. The narrow-leafed campion seeds mentioned earlier, germinated after 32,000 years, are the oldest seeds ever successfully revived, but they are likely not the oldest viable seeds in existence. There are seeds in the permafrost that may be even older, waiting for the thaw, waiting for the chance to grow, carrying genetic information from ecosystems that no longer exist, from climates that have vanished, from a world that has moved on without them. Seed banks, both natural and human-made, are archives of biodiversity. The Svalbard Global Seed Vault, buried in the permafrost of a Norwegian island in the Arctic, contains more than 1 million seed samples from around the world, a backup of global agriculture, a hedge against catastrophe. The vault is designed to remain frozen even without power, relying on the natural cold of the permafrost, and it is built to withstand earthquakes, nuclear war, and the rise of sea levels. Inside, seeds are stored in sealed foil packets at temperatures of -18 degrees Celsius, conditions that slow metabolic processes to nearly zero, extending viability for decades, centuries, potentially millennia. This is humanity's attempt to do what plants have been doing for millions of years, to preserve the future in the form of seeds, to wait out disasters, to ensure that life can continue. But natural seed banks, the seeds lying dormant in the soil, are far more extensive than any human archive. In a single square metre of soil in a temperate grassland, there may be tens of thousands of seeds, representing dozens of species, some freshly shed, others years or decades old. This is the seed bank of the soil, a reservoir of genetic diversity, a library of potential futures. When the soil is disturbed, by fire, by grazing, by ploughing, by flooding, dormant seeds are brought to the surface, exposed to light and oxygen, and they germinate, sometimes in species that have not been seen in the area for years. The landscape transforms, and the transformation is driven by seeds that were waiting, invisible beneath the surface. The longevity of seeds is not unlimited. Over time, even in ideal storage conditions, seeds accumulate damage. DNA degrades, proteins denature, membranes lose integrity. The rate of deterioration depends on temperature, moisture, and oxygen levels. Seeds stored in warm, humid conditions lose viability quickly, sometimes within months. Seeds stored in cool, dry conditions can remain viable for years or decades. Seeds stored frozen can remain viable for centuries or longer. But eventually, all seeds die. The embryo loses the ability to repair the accumulated damage, and when the seed is planted, nothing happens. The potential is gone, the future that was stored in the seed is lost, and the lineage ends, unless other seeds from the same plant have survived elsewhere. Some plants produce enormous numbers of seeds as a strategy to ensure that at least a few survive. A single orchid capsule can contain millions of seeds, each one tiny, dust-like, containing almost no stored nutrients, relying entirely on a partnership with a specific fungus to provide the energy needed for germination and early growth. The odds of any individual orchid seed finding the right fungus in the right location are vanishingly small, but the plant produces so many seeds that a few will succeed, and that is enough. This is the strategy of overwhelming the odds, of playing the lottery with millions of tickets, accepting that most will lose, but confident that some will win. Other plants produce very few seeds, but invest heavily in each one, provisioning it with abundant nutrients, encasing it in a hard shell, sometimes even providing it with a fleshy fruit to attract animals that will disperse it. The coconut is an extreme example, a seed so large and so well provisioned that it can float across oceans, surviving weeks or months in salt water, and then germinate on a distant beach, establishing a new population far from its parent. The coconut contains water, sugars, fats, and proteins, everything the embryo needs to grow a root system and a shoot capable of photosynthesis before the stored reserves are exhausted. This is the strategy of quality over quantity, of investing everything in a few well-equipped offspring, trusting that their superior provisioning will give them a better chance of survival. The dispersal of seeds is a story in itself, a story of partnerships between plants and animals, between plants and wind, between plants and water. Some seeds are equipped with wings or parachutes of fine hairs that allow them to drift on the wind, traveling miles from the parent plant. Dandelion seeds, milkweed seeds, maple samaras, all are designed to catch the air, to ride the currents, to colonize new ground. Other seeds are equipped with hooks or barbs that catch on the fur of passing animals, hitching a ride to new locations. Burdock seeds, cocklebur seeds, beggar's ticks, all are designed to attach, to be carried, to be deposited elsewhere, often far from where they were produced. Some seeds are dispersed by ants, which are attracted to a fatty appendage on the seed called an elaiosoma. The ant carries the seed back to its nest, eats the elaiosome, and discards the seed in the nest's refuse pile, which is rich in nutrients and provides an ideal germination site. This is myrmecochory, seed dispersal by ants, and it is common in many plant families, particularly in forests and woodlands. The plant provides food for the ant, and the ant provides dispersal and a nutrient-rich planting site for the seed, a mutualism that benefits both partners. Other seeds are dispersed by birds and mammals that eat the fruit and excrete the seeds, often far from the parent plant. The seeds of many fruit-bearing plants have hard coats that resist digestion, and passage through the animal's gut may actually enhance germination by scarifying the seed coat or by exposing the seed to digestive enzymes that break down chemical inhibitors. The animal gets nutrition from the fruit, and the plant gets dispersal and a deposit of fertilizer in the form of the animal's feces. This is a partnership millions of years old, and it has shaped the evolution of both plants and animals, leading to the bright colors and sweet flavors of fruits, which are advertisements directed at animals, signals that say, "Eat me and carry my seeds to new places." The seed is a time capsule, a message in a bottle, a bet on the future. It contains the genetic information of the parent plant the instructions for building a new individual, along with the resources to begin that construction. It is designed to wait, to endure, to survive conditions that would kill an actively growing plant. It is designed to disperse, to travel, to colonize new ground. And it is designed to germinate at the right time, in the right place, when the conditions are suitable for growth and reproduction. The seed is one of the most successful adaptations in the history of life on Earth, and it is the reason that plants have colonized nearly every terrestrial habitat, from the wettest rainforests to the driest deserts, from the equator to the poles, from sea level to the highest mountains. The seed waits, and in waiting, it endures, and in enduring, it ensures that the green world continues generation after generation across the vast stretches of time. Light falls on a leaf, and the world changes. Energy from a star 93 million miles away is captured, converted, woven into the fabric of life, and in that transformation, the atmosphere itself is remade, breath by breath, molecule by molecule, across billions of years. Photosynthesis is the most important chemical reaction on Earth, the foundation upon which nearly all life depends, and it happens in silence, in structures so small they are invisible to the naked eye, in organelles called chloroplasts that float in the cells of every green leaf, every blade of grass, every strand of algae in the ocean. Inside each chloroplast, stacked like coins, are the thylakoids, membrane-bound compartments containing chlorophyll, the green pigment that captures light. When a photon of light strikes a chlorophyll molecule, it excites an electron, boosting it to a higher energy state, and that electron is passed along a chain of proteins embedded in the thylakoid membrane, each transfer releasing a small amount of energy, which is used to pump hydrogen ions across the membrane, creating a gradient, a difference in concentration that stores potential energy, like water behind a dam. The hydrogen ions flow back through a protein called ATP synthase, which rotates like a turbine, using the energy of the flowing ions to synthesize ATP, the universal energy currency of cells. The excited electrons, meanwhile, are used to split water molecules, tearing them apart into hydrogen ions, electrons, and oxygen. The oxygen is released as a waste product, expelled through the stomata, the tiny pores on the leaf surface, into the atmosphere. This is the oxygen we breathe, the oxygen that makes up 21% of the atmosphere, and nearly all of it was produced by photosynthesis, by plants and algae and cyanobacteria over billions of years. Before photosynthesis evolved, the atmosphere contained almost no free oxygen. The early Earth was a reducing environment rich in methane and ammonia and hydrogen, and life, if it existed at all, was anaerobic, thriving in the absence of oxygen. Then, approximately 3 billion years ago, cyanobacteria evolved the ability to perform oxygenic photosynthesis, splitting water and releasing oxygen, and over the next billion years, oxygen accumulated in the atmosphere, transforming the planet, rusting the iron in the oceans and the rocks, creating the red beds and banded iron formations that geologists study today, and eventually reaching levels high enough to support aerobic respiration, the oxygen-dependent metabolism that powers most complex life. The carbon dioxide that plants absorb from the air is fixed into organic molecules through a cycle of reactions called the Calvin cycle, which takes place in the stroma, the fluid-filled space inside the chloroplast surrounding the thylakoids. The enzyme rubisco, the most abundant protein on Earth, catalyzes the first step, attaching a molecule of carbon dioxide to a 5-carbon sugar, which then splits into 2 3-carbon molecules. These molecules are processed through a series of steps, using the ATP and the reducing power generated by the light reactions. And eventually, they are assembled into glucose, a 6-carbon sugar that serves as the building block for all the other organic molecules the plant needs: cellulose for cell walls, starch for energy storage, amino acids for proteins, lipids for membranes. The plant is building itself from air and water and light, constructing complex molecules from simple ones, increasing order in a universe that tends toward disorder, and it does this continuously as long as the sun is shining and water is available. A single leaf, no larger than your hand, can contain tens of millions of chloroplasts, and each chloroplast can perform thousands of photosynthetic reactions per second. Scaled up to the level of a tree, a forest, a biome, the numbers become staggering. A large oak tree, with tens of thousands of leaves, can fix hundreds of kilograms of carbon per year, pulling carbon dioxide from the atmosphere and locking it into wood, into the cellulose and lignin that make up the trunk and branches, which can stand for centuries, storing that carbon, keeping it out of the atmosphere. Forests are carbon sinks, absorbing more carbon than they release, and the Amazon rainforest alone contains approximately 150 billion metric tons of carbon stored in the living and dead biomass, in the trees and the soil and the leaf litter. When forests are cleared, that carbon is released either through burning or through decomposition, and it returns to the atmosphere as carbon dioxide, contributing to the greenhouse effect, to the warming of the planet. The efficiency of photosynthesis varies depending on the pathway the plant uses. Most plants use the C3 pathway, named for the 3-carbon molecule that is the first stable product of carbon fixation. C3 photosynthesis is efficient in cool, moist environments, but it has a weakness. Rubisco, the enzyme that fixes carbon dioxide, can also react with oxygen, a process called photorespiration, which wastes energy and reduces the net carbon gained. In hot, dry environments where plants close their stomata to conserve water, the concentration of oxygen inside the leaf rises, and the concentration of carbon dioxide falls, and photorespiration becomes a significant problem. To overcome this, some plants have evolved alternative pathways. C4 plants, which include corn, sugarcane, and many tropical grasses, have a modified anatomy and biochemistry that concentrates carbon dioxide around rubisco, minimizing photorespiration. They fix carbon dioxide in the mesophyll cells using a different enzyme, PEP carboxylase, which produces a 4-carbon molecule. This molecule is transported to specialized bundle sheath cells where it is broken down, releasing carbon dioxide in high concentrations, which is then fixed by rubisco in the normal Calvin cycle. This spatial separation of the initial carbon fixation and the Calvin cycle allows C4 plants to photosynthesize efficiently even when stomata are partially closed, making them well suited to hot, dry environments. C4 photosynthesis is more energy intensive than C3, requiring more ATP, but in the right conditions the reduction in photorespiration more than compensates, and C4 plants can achieve higher growth rates and higher water use efficiency than C3 plants. Cam plants, which include cacti, succulents, and some orchids, take the strategy even further. CAM stands for crassulacean acid metabolism, named for the family Crassulaceae, where it was first discovered. CAM plants open their stomata at night when temperatures are cooler and humidity is higher, and they fix carbon dioxide into organic acids, which are stored in the vacuoles of the cells. During the day, the stomata close, preventing water loss, and the stored acids are broken down, releasing carbon dioxide inside the leaf, where it is fixed by rubisco in the Calvin cycle. This temporal separation of carbon dioxide uptake and fixation allows CAM plants to survive in extremely arid environments, where water is scarce and evaporation rates are high. A CAM plant can have water use efficiency 10 times higher than a C3 plant, losing only a fraction of the water per unit of carbon fixed. The light that drives photosynthesis is not uniform. Some light is a mixture of wavelengths, from ultraviolet to infrared, and different wavelengths carry different amounts of energy. Blue light with shorter wavelengths carries more energy per photon than red light with longer wavelengths. Chlorophyll absorbs blue and red light most efficiently, and it reflects green light, which is why plants appear green. But there are other pigments in the chloroplast. Carotenoids, which absorb blue and green light and appear yellow or orange, and phycobilins in cyanobacteria and red algae, which absorb green and yellow light and appear red or blue. These accessory pigments capture wavelengths that chlorophyll cannot, and they transfer the energy to chlorophyll, broadening the spectrum of light that can be used for photosynthesis. In the fall, when chlorophyll breaks down and is not replaced, the carotenoids become visible and the leaves turn yellow and orange, a final display of colour before the leaves are shed. The amount of light a plant receives varies with the time of day, the season, the weather, and the position of the plant in the canopy. Plants in full sun receive intense light, sometimes more than they can use, and they must dissipate the excess energy to avoid damage to the photosynthetic machinery. They do this through a process called non-photochemical quenching, in which excess energy is released as heat, mediated by changes in the structure of the thylakoid membrane and the activation of specific proteins. Plants in deep shade receive very little light, and they must maximize their light capture, producing more chlorophyll, enlarging their leaves, orienting their leaves to face the light. Shade leaves are typically thinner, darker green, and have a larger surface area than sun leaves, adaptations that allow them to capture as much of the limited light as possible. Some plants can track the sun across the sky, a phenomenon called heliotropism. Sunflowers are the most famous example. The young flower heads face east in the morning, and they slowly rotate to follow the sun across the sky, facing west by evening. At night, they rotate back to face east, ready for the sunrise. This movement is driven by differential growth rates on opposite sides of the stem, controlled by hormones and by the plant's internal circadian clock. The advantage of heliotropism is that it maximizes light capture, keeping the flower head perpendicular to the sun's rays throughout the day. Interestingly, once the sunflower matures and the flower head is fully developed, the tracking stops and the flower remains facing east. This eastward orientation warms the flower head in the morning sun, which attracts more pollinators, particularly bees, which are more active in warmer conditions. The stomata, the pores on the leaf surface through which carbon dioxide enters and oxygen and water vapor exit, are marvels of biological engineering. Each stoma is surrounded by two guard cells which can change shape, opening and closing the pore in response to environmental signals. When the guard cells take up water, they swell and curve, opening the stoma. When they lose water, they shrink and straighten, closing the stoma. The opening and closing are controlled by the movement of potassium ions and other solutes into and out of the guard cells, which changes the osmotic pressure and drives water movement. Stomata typically open in the morning, when light is available for photosynthesis, and close at night, when photosynthesis stops. They also close in response to water stress, to high temperatures, to high carbon dioxide concentrations inside the leaf, and to the hormone abscisic acid, which is produced when the plant is under stress. The density of stomata varies widely among species, and even within a single plant, depending on the environment. Plants in dry environments typically have fewer stomata, reducing water loss. Plants in wet environments can afford to have more stomata, maximizing carbon dioxide uptake. Some plants have stomata only on the lower surface of the leaf, which reduces water loss because the lower surface is shaded and cooler. Other plants have stomata on both surfaces, which increases gas exchange, but also increases water loss. The size and density of stomata are not fixed. They can change during development in response to environmental conditions, a form of phenotypic plasticity that allows the plant to optimize its performance in different environments. The water that evaporates from the leaves, a process called transpiration, is not wasted. Transpiration drives the movement of water from the roots to the leaves, pulling water up through the xylem vessels in a continuous column, powered by the evaporation at the leaf surface. This is the transpiration stream, and it carries not only water but also dissolved minerals from the soil: nitrogen, phosphorus, potassium, calcium, magnesium, and trace elements that the plant needs for growth. Transpiration also cools the leaf, preventing overheating in bright sunlight. A large tree can transpire hundreds of liters of water per day, and this water, released into the atmosphere, contributes to cloud formation and precipitation, linking the plant to the water cycle, to the climate, to the weather patterns of entire regions. The relationship between photosynthesis and respiration is a cycle, a balance. During the day, photosynthesis exceeds respiration, and the plant produces more oxygen than it consumes, more sugars than it burns. At night, photosynthesis stops, but respiration continues, and the plant consumes oxygen and releases carbon dioxide, burning sugars to power its metabolism, its growth, its maintenance. Over a 24-hour period, a healthy plant produces more oxygen and fixes more carbon than it consumes, resulting in net growth, net carbon storage. But the balance is delicate, and it can shift. A plant under stress, a plant in deep shade, a plant that is diseased or damaged may respire more than it photosynthesizes, resulting in net carbon loss, in decline, in eventual death. Photosynthesis is ancient, but it is not static. It is still evolving, still adapting, still responding to the changing environment. The rise in atmospheric CO2 due to human activity is changing the conditions for photosynthesis, and plants are responding. Some plants are growing faster, producing more biomass because carbon dioxide is the raw material for photosynthesis, and more carbon dioxide means more potential for growth, at least up to a point. But the response is not uniform. C3 plants benefit more than C4 plants, because C4 plants are already efficient at concentrating carbon dioxide, and the benefits are limited by other factors: water, nutrients. A plant cannot grow indefinitely faster simply because there is more carbon dioxide. It needs water to open its stomata. It needs nitrogen to build proteins. It needs phosphorus for DNA and ATP, and if any of these is limiting, growth will be constrained. The green world is a solar-powered engine, capturing the energy of the sun and converting it into the chemical energy that fuels nearly all life on Earth. Every breath you take, every bite of food you eat, every movement you make is powered ultimately by photosynthesis, by the light falling on leaves, by the quiet work of chloroplasts, by the ancient partnership between life and light that has shaped the planet for billions of years and will continue to shape it for billions more, as long as the sun shines and the green world endures. Plants speak to one another in a language older than words, a vocabulary of volatile molecules drifting on the breeze, carrying warnings of danger, invitations to allies, and messages we are only beginning to decode. When a caterpillar chews into a leaf, it does more than create a hole. The mechanical damage, the tearing of cell walls, triggers a cascade of chemical responses within the plant, a defensive mobilization that begins within seconds and spreads throughout the entire organism within minutes. The damaged cells release enzymes that break down membrane lipids, producing jasmonic acid, a hormone that acts as an alarm signal traveling through the plant's vascular system and activating genes involved in defense. Within hours, the plant begins producing compounds that make its tissues less palatable, less nutritious, harder to digest. Tannins accumulate in the leaves, binding to proteins and reducing their digestibility. Protease inhibitors are synthesized, molecules that interfere with the digestive enzymes in the caterpillar's gut, making it harder for the insect to extract nutrients from the plant tissue. The caterpillar, encountering these defenses, eats less, grows more slowly, becomes more vulnerable to predators and parasites. But the plant does not stop there. It also releases volatile organic compounds into the air: green leaf volatiles, terpenoids, methyl jasmonate, molecules that evaporate from the damaged tissue and drift on the wind. These airborne chemicals serve multiple functions. They warn neighboring plants, even plants of different species, that herbivores are present, and the neighboring plants, detecting these volatiles, begin producing their own defensive compounds before they are attacked. This is preemptive defense, a form of eavesdropping, of learning from the misfortune of others. The volatiles also attract predators and parasitoids, insects that prey on or parasitize the herbivores. A tobacco plant attacked by caterpillars releases a specific blend of volatiles that attracts parasitic wasps, which lay their eggs inside the caterpillars. The wasp larvae hatch and consume the caterpillar from the inside, killing it. The plant is recruiting bodyguards, calling for help in a chemical language that the wasps understand and respond to. The specificity of these signals is remarkable. A plant can distinguish between different types of damage. Chewing by caterpillars produces a different chemical signature than sucking by aphids, and the plant tailors its response accordingly. Caterpillar damage triggers the production of volatiles that attract wasps. Aphid damage triggers the production of volatiles that attract ladybugs and lacewings, predators of aphids. The plant is not responding blindly. It is identifying the threat and calling for the appropriate allies. This requires a level of perception and discrimination that challenges our traditional understanding of what it means to be a plant, what it means to be intelligent, what it means to communicate. The chemical communication between plants is not limited to warnings of herbivore attack. Plants also communicate about water stress, about nutrient availability, about the presence of pathogens. A plant experiencing drought will produce abscisic acid, which closes the stomata to conserve water. And some of this abscisic acid may be released into the soil, where it can be detected by neighboring plants, which then begin conserving water themselves, preparing for the drought before it affects them directly. A plant infected by a fungal pathogen will produce salicylic acid, another hormone that activates immune responses, and volatiles derived from salicylic acid may warn neighboring plants to bolster their own defenses. The air and the soil are filled with these chemical messages, a constant exchange of information that shapes the behavior and the physiology of the entire plant community. Some plants release allelopathic chemicals, compounds that inhibit the growth of competing plants. Black walnut trees produce juglone, a toxic compound that is released from the roots and the decomposing leaves, and it accumulates in the soil, preventing the germination and growth of many other plant species. This gives the walnut tree a competitive advantage, reducing competition for water, nutrients, and light. The area beneath a black walnut tree is often bare or sparsely vegetated, a chemical exclusion zone maintained by the tree's allelopathic compounds. Other plants produce allelopathic compounds that are released into the air. Sagebrush, common in the arid regions of the western United States, releases volatile terpenoids that inhibit the germination of nearby plants, particularly grasses, reducing competition and maintaining the dominance of sagebrush in the ecosystem. The mycorrhizal network, the underground fungal web connecting plant roots, is also a conduit for chemical communication. When a plant is attacked by aphids, it can send chemical signals through the mycorrhizal network to neighboring plants, which then increase their production of defensive enzymes. This has been demonstrated experimentally with broad beans connected by mycorrhizal fungi. When one plant was infested with aphids, the connected plants showed increased levels of defensive compounds, even though they had not been attacked themselves. The signal traveled through the fungal network, through the hyphae connecting root to root, a subterranean telegraph system carrying warnings of danger. Plants can also communicate with their own future selves. Some plants produce defensive compounds in response to herbivore attack, and these compounds persist in the tissues for weeks or months, providing protection against future attacks. This is induced resistance, a form of immunological memory, and it can be systemic, spreading throughout the plant from the site of the initial attack. A plant that has been attacked once is better defended against subsequent attacks, and this heightened state of defense can last for the entire growing season. The plant remembers the attack, not in the way an animal remembers, with neurons and synapses, but in the altered expression of genes, in the elevated levels of defensive proteins, in the primed state of the immune system, ready to respond more quickly and more strongly to the next threat. The roots of plants release exudates, a complex mixture of sugars, amino acids, organic acids, and secondary metabolites into the surrounding soil. These exudates serve multiple functions. They attract beneficial microbes, bacteria, and fungi that help the plant acquire nutrients, that protect it from pathogens, that stimulate its growth. They also deter harmful microbes, creating a chemical barrier around the root. The composition of the exudates varies depending on the plant species, the developmental stage, the nutritional status, and the presence of stress. A plant deficient in phosphorus will release exudates that solubilize phosphorus in the soil, making it more available for uptake. A plant under attack by root-feeding nematodes will release exudates that attract nematode-trapping fungi, which capture and kill the nematodes. The root is actively shaping the microbial community of the rhizosphere, cultivating allies, deterring enemies, through the strategic release of chemical signals. Some plants can even detect the presence of neighboring plants and adjust their growth accordingly. This is called kin recognition, and it has been demonstrated in several species. When a plant is grown next to a sibling, a plant from the same parent, it allocates fewer resources to root growth and more to shoot growth, reducing competition with the sibling. When the same plant is grown next to an unrelated individual, it allocates more resources to root growth, competing more aggressively for soil resources. The plant is distinguishing kin from non-kin, presumably through chemical signals in the soil, through the exudates released by the roots, and it is adjusting its competitive strategy accordingly. This suggests a level of social behavior in plants, a capacity to recognize relatives and to cooperate with them, that was entirely unexpected. The volatile compounds released by plants are not only signals, they are also defenses in their own right. Many terpenoids, the most common class of plant volatiles, are toxic or repellent to insects and other herbivores. The strong scent of mint, of rosemary, of lavender is due to terpenoids, and these scents deter many herbivores. The plant is not just calling for help, it is also directly defending itself, making itself less attractive, less palatable, harder to find. The volatiles can also have antimicrobial properties, inhibiting the growth of bacteria and fungi on the leaf surface, protecting the plant from infection. The timing of volatile release is often synchronized with the activity patterns of pollinators or herbivores. Many flowers release their strongest scents at the time of day when their pollinators are most active. Night-blooming flowers, pollinated by moths, release their scents at dusk and during the night. Day-blooming flowers, pollinated by bees, release their scents during the day. The plant is advertising its presence to the right audience at the right time, maximizing the efficiency of pollination. Similarly, some plants release defensive volatiles primarily during the day, when herbivorous insects are most active, and reduce or stop the release at night, conserving resources. The chemical communication of plants is not a recent discovery, but our understanding of its complexity and its importance has grown dramatically in recent decades. We now know that plants are not passive, silent organisms, but active participants in a rich and complex chemical dialogue, communicating with each other, with insects, with microbes, shaping their environment and responding to it in ways that are sophisticated, nuanced, and adaptive. The air in a forest, in a meadow, in a field of crops, is thick with chemical messages, a conversation conducted in a language of molecules, a language that we are only beginning to learn to read, a language that has been spoken for hundreds of millions of years, long before humans existed, long before any creature capable of speech walked the earth. The implications of this chemical communication are profound. It means that plants are not isolated individuals, but members of communities, connected by networks of chemical signals both above ground and below ground. It means that the health and the productivity of a plant depend not only on its own genetics and its own physiology, but also on its interactions with its neighbours, with the microbes in the soil, with the insects in the air. It means that agriculture, forestry, and conservation must take into account these interactions, these chemical dialogues. If we are to manage ecosystems sustainably, if we are to understand how plants respond to stress, to climate change, to the presence of pests and pathogens, the silent communication of plants is not silent at all. It is a constant dynamic exchange of information, shaping the green world in ways we are only beginning to appreciate, in ways that remind us that intelligence, communication, and awareness are not the exclusive province of animals, but are woven into the fabric of life itself in forms diverse and ancient and endlessly surprising. Water rises against gravity in the tallest trees on Earth, climbing more than 300 feet from root to leaf, powered by no pump, no heart, no moving parts, only the elegant physics of cohesion, adhesion, and the patient pull of evaporation. The coast redwoods of California, Sequoia sempervirens, are the tallest living organisms on Earth, reaching heights that exceed 380 feet, taller than the Statue of Liberty, taller than a 30-story building. Standing at the base of one of these giants, looking up through the canopy into the filtered green light, you are confronted with a hydraulic puzzle. How does water reach the topmost leaves, hundreds of feet above the ground, against the force of gravity, through a column of dead cells without any mechanical pump to push it upward? The answer lies in the properties of water itself, in the hydrogen bonds that link water molecules to one another, and in the structure of the xylem, the vascular tissue that transports water from roots to leaves. Water molecules are polar, with a slight positive charge on the hydrogen atoms and a slight negative charge on the oxygen atom, and this polarity allows them to form hydrogen bonds with one another, weak bonds that are constantly forming and breaking, but collectively strong enough to hold water molecules together in a continuous column. This is cohesion, the attraction of water molecules to one another, and it is what allows water to be pulled upward through the xylem without breaking apart. The xylem vessels are narrow tubes, only micrometers in diameter, and the water inside them is under tension, under negative pressure, pulled upward by the evaporation of water from the leaves. This is the cohesion tension theory, and it explains how water can rise to heights that would be impossible with any mechanical pump. The xylem vessels themselves are remarkable structures. They are composed of dead cells, cells that have lost their cytoplasm and their nuclei, leaving only the cell walls, which are reinforced with lignin, a complex polymer that makes the walls rigid and strong, able to withstand the negative pressure without collapsing. The cells are stacked end to end, and the end walls are perforated or completely dissolved, creating a continuous tube from root to leaf. In angiosperms, flowering plants, these tubes are called vessels, and they can be several meters long. In gymnosperms, conifers like the redwoods, the tubes are called tracheids, and they are shorter, with water moving from one tracheid to the next through pits, small openings in the cell walls. Tracheids are less efficient than vessels at conducting water, but they are also safer, less prone to cavitation, the formation of air bubbles that can break the water column and block water transport. Cavitation is the greatest threat to the hydraulic system of a tree. When the tension in the xylem becomes too great, when the negative pressure exceeds a critical threshold, the water column can break and an air bubble, an embolism, forms in the vessel. Once an embolism forms, that vessel is no longer functional. It cannot conduct water. If too many vessels become embolized, the tree can no longer transport enough water to the leaves, and the leaves wilt, the photosynthesis stops, and the tree dies. Cavitation is more likely to occur during drought, when the soil is dry and the tension in the xylem is highest, and it is also more likely in vessels with larger diameters, because larger vessels are more vulnerable to the formation of air bubbles. This is why conifers, which live in environments where drought and freezing are common, have tracheids instead of vessels. Tracheids are narrower and safer, less efficient but more reliable. The redwoods have evolved strategies to minimize the risk of cavitation. Their tracheids are narrow, and the pits connecting one tracheid to the next are small and reinforced, preventing air bubbles from spreading from one tracheid to another. The redwoods also have the ability to refill embolized tracheids, dissolving the air bubbles and restoring water transport, though the mechanisms are not fully understood, and the redwoods have another source of water, one that is unique among trees. They absorb water directly from fog through their leaves. The coast of Northern California is frequently shrouded in fog, especially during the summer, and the redwoods intercept this fog, condensing it on their needles and absorbing it through the stomata and through the cuticle, the waxy layer on the leaf surface. This foliar water uptake can provide a significant fraction of the tree's water needs, reducing the demand on the root system and reducing the tension in the xylem, making cavitation less likely. The roots of a redwood are surprisingly shallow for such a tall tree. They rarely extend more than 10 or 12 feet deep, but they spread laterally, covering an area that can exceed an acre, and they intertwine with the roots of neighboring trees, creating a network of mutual support. The shallow roots are an adaptation to the wet, poorly drained soils of the coastal forests, where deep roots would be submerged and oxygen-starved. The lateral spread and the intertwinement provide stability, anchoring the tree against the strong winds that sweep in from the Pacific Ocean. A redwood standing alone is vulnerable to wind throw, to being toppled by the wind. But a redwood in a grove, surrounded by other redwoods, their roots interlocked, is remarkably stable, able to withstand storms that would fell isolated trees. The root system is not just an anchor, it is also a foraging organ, constantly growing, exploring the soil, seeking water and nutrients. The root tips, the very ends of the growing roots, are covered with root hairs, tiny extensions of the epidermal cells that increase the surface area of the root by a factor of 10 or more, allowing the root to absorb water and nutrients more efficiently. The root hairs are short-lived, lasting only a few days or weeks, but they are constantly being replaced, and at any given time, a mature tree may have billions of root hairs, a vast absorptive surface in contact with the soil. The roots also form partnerships with mycorrhizal fungi, which extend the reach of the root system even further, accessing water and nutrients that the roots alone could never reach. The transport of water from the roots to the leaves is only half of the story. The tree must also transport sugars, the products of photosynthesis, from the leaves to the rest of the tree, to the roots, the trunk, the branches, the developing seeds. This is the function of the phloem, the other major vascular tissue. Unlike the xylem, which is composed of dead cells, the phloem is composed of living cells, called sieve tube elements, which are connected end to end to form sieve tubes. The end walls of the sieve tube elements are perforated with sieve plates, which allow the flow of sugars and other organic molecules from one cell to the next. The sieve tube elements have lost their nuclei and most of their organelles, but they are still alive, still metabolically active, and they are supported by companion cells, which retain their nuclei and provide the sieve tube elements with the proteins and the energy they need to function. The movement of sugars through the phloem is driven by pressure flow, a process fundamentally different from the tension-driven flow of water in the xylem. Sugars are loaded into the sieve tubes in the leaves, increasing the solute concentration and drawing water into the sieve tubes by osmosis. This creates a positive pressure, a turgor pressure, that pushes the sugars and the water through the sieve tubes toward the roots and other sink tissues, where the sugars are unloaded and used for growth, for storage, for respiration. The unloading of sugars decreases the solute concentration in the sieve tubes, and water moves out by osmosis, reducing the pressure. The difference in pressure between the source, the leaves, and the sink, the roots, drives the flow of sugars through the phloem, a flow that can be remarkably fast, moving at rates of up to a meter per hour. The phloem is vulnerable to damage. Because the sieve tube elements are living cells, they can be killed by freezing, by heat, by pathogens, by herbivores. Aphids and other sap-sucking insects feed on the phloem, inserting their stylets into the sieve tubes and withdrawing the sugar-rich sap. A heavy infestation of aphids can weaken a tree, reducing its growth, making it more vulnerable to other stresses. Bark beetles bore through the bark and feed on the phloem, and they also introduce fungal pathogens that can block the phloem, cutting off the flow of sugars to the roots. If the phloem is girdled, if a continuous ring of bark is removed from the trunk, the tree will die, because the roots will be starved of sugars, and without the roots, the tree cannot absorb water and nutrients. The cambium, the thin layer of cells between the xylem and the phloem, is the growth engine of the tree. The cambium cells divide continuously during the growing season, producing new xylem cells to the inside and new phloem cells to the outside, adding girth to the trunk and the branches. The cambium is sensitive to hormones, to temperature, to the availability of water and nutrients, and it adjusts its activity accordingly. In the spring, when water is abundant and temperatures are rising, the cambium produces large, thin-walled xylem cells optimized for water transport. In the summer, when water is less abundant and growth slows, the cambium produces smaller, thicker-walled cells optimized for strength. The transition from the large cells of spring to the small cells of summer creates the visible growth ring, and by counting the rings, we can determine the age of the tree, and by measuring the width of the rings, we can infer the growing conditions of each year. The oldest redwoods are more than 2,000 years old. They were seedlings when the Roman Empire was at its height, saplings when the Library of Alexandria was burned, mature trees when the first European explorers reached the coast of California. They have survived droughts, floods, fires, earthquakes, and the slow grinding of geological time. Their longevity is a testament to their resilience, to their ability to repair damage, to resist disease, to endure. The heartwood of a redwood, the inner core of the trunk, is saturated with tannins and other compounds that make it resistant to decay, resistant to insects, resistant to fire. A redwood can be hollowed out by fire, burned so severely that you can stand inside the trunk, and yet the tree survives because the living tissue, the cambium and the phloem and the sapwood, the outer layers of xylem, are still intact, still functional, still transporting water and sugars, still growing. The redwoods are not the only trees with impressive hydraulic systems. The giant sequoias, Sequoia dendron giganteum, are shorter than the coast redwoods, but more massive, with trunks that can exceed 30 feet in diameter and weights that can exceed 2 million pounds. The eucalyptus trees of Australia can reach heights of over 300 feet, and they transpire enormous quantities of water, drawing down the water table and drying out the soil. The baobabs of Africa have massive trunks that store water, allowing them to survive long dry seasons. Each species has evolved its own solutions to the hydraulic challenges of being a tree, of lifting water against gravity, of balancing water loss with water uptake, of surviving drought and flood and fire. The hydraulic architecture of a tree is not fixed. It is dynamic, responsive, adaptive. A tree can adjust the diameter of its xylem vessels, the density of its stomata, the depth and the spread of its roots, in response to the environment, in response to stress, in response to the availability of water. A tree growing in a dry environment will produce narrower vessels, reducing the risk of cavitation, and it will produce deeper roots, accessing water that is unavailable to shallow-rooted plants. A tree growing in a wet environment can afford to produce wider vessels, maximizing water transport, and it can produce shallower roots because water is abundant near the surface. This phenotypic plasticity, this ability to adjust the body plan in response to the environment, is one of the keys to the success of trees, to their ability to colonize a wide range of habitats, from the wettest rainforests to the driest deserts, from the equator to the edge of the Arctic. The water that moves through a tree is not just water, it is a solution, carrying dissolved minerals from the soil, nitrogen and phosphorus, and potassium and calcium and magnesium, and trace elements, all the nutrients the tree needs to build proteins and nucleic acids and chlorophyll and cell walls. The concentration of these nutrients in the xylem sap is low, typically less than 1%, but the volume of water moving through the tree is so large that the total amount of nutrients transported is substantial. A large tree can transport kilograms of nitrogen per year, kilograms of phosphorus, kilograms of potassium, all dissolved in the water flowing from roots to leaves, all essential for growth, for photosynthesis, for reproduction. The redwoods stand as monuments to the power of water, to the physics of cohesion and adhesion, to the elegance of biological engineering. They are living proof that life can overcome gravity, can build structures of astonishing height and complexity, can endure for millennia using nothing more than sunlight and water and air and the minerals of the soil. They are reminders that the natural world is full of wonders, of solutions to problems that seem insurmountable, of beauty and resilience and patience. And they invite us to slow down, to look up, to marvel at the quiet achievements of the green world, achievements that unfold on timescales we are not accustomed to perceiving, in processes we are only beginning to understand, in a language of physics and chemistry and biology that speaks to the fundamental unity of life, to the deep connections that link all living things, from the smallest bacterium to the tallest tree. When the sun sets and the human world quiets, the green world does not simply stop, it shifts, entering a different state, a nocturnal rhythm governed by internal clocks older than the moon, older than the stars we navigate by. Plants sleep, though not in the way animals sleep. They do not close their eyes because they have no eyes. They do not enter REM sleep because they have no brain. But they do enter a state of rest, a state of reduced activity, a state governed by circadian rhythms, internal biological clocks that cycle with a period of approximately 24 hours, synchronized to the daily cycle of light and dark. These rhythms are not imposed from outside. They are generated from within by networks of genes and proteins that oscillate, turning on and off in regular cycles, driving changes in metabolism, in growth, in the opening and closing of stomata, in the folding and unfolding of leaves. Even in constant darkness, even in constant light, the rhythms persist, proof that they are endogenous, internal, a fundamental property of the plant's biology. The most visible manifestation of plant sleep is nyctimasty, the sleep movement of leaves. Many plants fold their leaves at night, a movement driven by changes in turgor pressure in specialized cells at the base of the leaf or the leaflet, called pulvini. During the day, the The pulvini on one side of the leaf base are swollen with water, holding the leaf open, horizontal, exposed to the light. At night, water moves out of these cells and into the pulvini on the opposite side, and the leaf folds, droops, closes. The movement is slow, taking minutes or hours, but it is deliberate, controlled, reversible. In the morning, the process reverses and the leaves open again, ready to capture the light. The function of nyctinasty is not entirely clear, but it may reduce heat loss at night, or reduce water loss through transpiration, or protect the leaves from herbivores that are active at night, or reduce damage from frost. The prayer plant, Maranta locunura, is famous for its dramatic nyctinastic movements. During the day, its leaves lie flat, displaying their intricate patterns of green and purple. At night, the leaves fold upward, like hands pressed together in prayer, and in the morning they unfold again. The movement is so regular, so predictable, that you can set your watch by it, and it continues even in constant conditions, even in a laboratory with artificial light on a fixed schedule. Proof that the movement is driven by an internal clock, not merely by the presence or absence of light. The circadian clock in plants is a complex network of genes that regulate each other in feedback loops. The core of the clock consists of genes that are expressed in the morning, genes that are expressed in the evening, and genes that are expressed at night. And these genes encode proteins that regulate the expression of other genes, creating oscillations that cycle with a period of approximately 24 hours. The clock is synchronized to the external environment by light, detected by photoreceptors in the plant, proteins that change shape when they absorb light, triggering signaling cascades that reset the clock, adjusting its phase to match the day-night cycle. This synchronization is essential because it allows the plant to anticipate the dawn, to prepare for photosynthesis before the sun rises, to open its stomata in the morning when light is available, to close them at night when photosynthesis stops. The circadian clock regulates not only sleep movements, but also a wide range of physiological processes. Photosynthesis is more efficient in the morning, when the enzymes of the Calvin cycle are most active, primed by the circadian clock. Growth is faster at night, when the plant is not investing energy in photosynthesis, and can allocate more resources to cell division and cell expansion. The production of defensive compounds is often higher at certain times of day, timed to coincide with the activity patterns of herbivores. The opening of flowers, the release of scent, the production of nectar are all regulated by the circadian clock, timed to match the activity of pollinators. The clock is a master regulator coordinating the plant's activities with the rhythms of the environment, optimizing performance, maximizing fitness. Some plants bloom only at night, opening their flowers as the sun sets and closing them at dawn. These night-blooming plants are typically pollinated by moths, bats, or other nocturnal animals, and their flowers are often white or pale colored, making them more visible in the darkness, and they release strong, sweet scents that attract their pollinators from a distance. The night-blooming cereus, a cactus native to the deserts of the southwestern United States and Mexico, produces enormous white flowers that open for a single night, releasing a fragrance that has been described as intoxicating, overwhelming, unforgettable. The flowers open at dusk, and by dawn they have wilted, never to open again. The plant invests enormous resources in these flowers, in the petals, the scent, the nectar, all for a single night of pollination, a single chance to reproduce. The moonflower, Ipomea alba, is a vine related to the morning glory, but unlike its diurnal cousin, the moonflower opens its flowers at night. The flowers are large, white, trumpet-shaped, and they release a scent that attracts hawk moths, which hover in front of the flowers, extending their long proboscises to reach the nectar at the base of the floral tube. The moths, dusted with pollen from previous flowers, transfer the pollen to the stigma, affecting pollination, and the cycle continues. The moonflower is native to tropical America, but it has been cultivated in gardens around the world, prized for its nocturnal beauty, for the way its flowers glow in the moonlight, for the way it transforms the night garden into a place of mystery and fragrance. The baobab trees of Africa and Madagascar produce large pendulous flowers that open at dusk and are pollinated by bats. The flowers hang from long stalks, making them accessible to the bats, which are attracted by the flowers' musky scent and their abundant nectar. The bats visit the flowers, lapping up the nectar, and in the process they transfer pollen from flower to flower, ensuring cross-pollination. By dawn the flowers have withered, and the bats have returned to their roosts, and the tree waits for the next night, the next opportunity for pollination. Not all nocturnal plant activity is related to pollination. Some plants release volatile compounds at night, compounds that deter herbivores or attract predators of herbivores. Some plants grow faster at night, when temperatures are cooler and humidity is higher, reducing water stress. Some plants absorb dew through their leaves at night, supplementing their water supply. The night is not a time of inactivity for plants. It is a time of different activities, of processes that are optimized for the absence of light, for the cooler temperatures, for the higher humidity, for the presence of nocturnal pollinators and nocturnal herbivores. The study of plant circadian rhythms has revealed that plants have a sense of time, a capacity to measure the passage of hours, to anticipate the future, to prepare for events that have not yet happened. A plant that is exposed to a short day, a day with less than a certain number of hours of light, will begin the process of flowering, even though the actual flowering may not occur for weeks or months. The plant is measuring the length of the day, comparing it to an internal threshold, and making a developmental decision based on that measurement. This is photoperiodism, the response to day length, and it is mediated by the circadian clock, which measures the duration of light and dark periods and integrates that information over time. Some plants are short-day plants, flowering when the days are shorter than a critical length, typically in the fall or winter. Chrysanthemums, poinsettias, and soybeans are short-day plants. Other plants are long-day plants, flowering when the days are longer than a critical length, typically in the spring or summer. Lettuce, spinach, and wheat are long-day plants. Still other plants are day-neutral, flowering regardless of day length, responding instead to other cues like temperature or plant age. Tomatoes, cucumbers, and roses are day-neutral plants. The photoperiodic response is an adaptation to seasonal environments, ensuring that flowering occurs at the time of year when conditions are most favorable for pollination and seed development. The molecular mechanisms of photoperiodism are complex, involving interactions between the circadian clock, photoreceptors that detect light, and genes that regulate flowering. In the model plant Arabidopsis, a long-day plant, a gene called constans is expressed in the afternoon, and its protein product accumulates in the evening. If the evening coincides with light, as it does in long days, the constans protein is stable, and it activates the expression of a gene called flowering locus T, which encodes a protein that moves from the leaves to the shoot apex, where it triggers the transition from vegetative growth to flowering. If the evening coincides with darkness, as it does in short days, the constans protein is degraded, and flowering is not triggered. The plant is using the circadian clock to measure the time of day, and it is using light to determine whether that time of day coincides with the presence of light. And from these two pieces of information, it infers the length of the day and makes a decision about whether to flower. The precision of the circadian clock is remarkable. In laboratory experiments, plants can distinguish between days that differ in length by as little as 15 minutes, and they can maintain accurate time for weeks or months in constant conditions. The clock is not a simple timer. It is a sophisticated information processing system, integrating multiple inputs, adjusting its phase and its period in response to environmental cues, and driving a wide range of outputs from gene expression to metabolism to growth to development. The clock is essential for the plant's survival, for its ability to compete, to reproduce, to adapt to changing environments. Plants also respond to the moon, to the lunar cycle, though the mechanisms are less well understood than the circadian clock. Some marine algae synchronize their reproduction with the lunar cycle, releasing gametes at specific phases of the moon, ensuring that fertilization occurs when tides are favorable for dispersal. Some terrestrial plants show lunar rhythms in growth or in the opening of flowers, though the adaptive significance of these rhythms is unclear. The moon provides much less light than the sun, and it is not obvious how plants detect the lunar cycle, but the correlations are real, documented in multiple species, suggesting that plants are sensitive to subtle environmental cues that we are only beginning to appreciate. The night is not the absence of the day, it is a different world, a world of different rhythms, different activities, different opportunities. For plants, the night is a time of rest, but also a time of growth, a time of preparation, a time of communication with nocturnal pollinators and nocturnal herbivores. The night is governed by the circadian clock, by the internal rhythms that cycle with the turning of the earth, by the ancient partnership between life and time, a partnership that has shaped the evolution of plants for billions of years and continues to shape them today in ways subtle and profound, in ways that remind us that the green world is always active, always responsive, always alive, even in the darkness, even in the silence, even when we are not watching. The green world is not defenseless. Beneath the beauty of petals and the elegance of leaves lies an arsenal of chemical weapons, physical barriers, and deceptive strategies refined over millions of years of evolutionary warfare. Plants cannot run from their enemies. Rooted in place, they must endure whatever comes, whether herbivore or pathogen, drought or fire. And so they have evolved defenses, layers upon layers of protection, some obvious, some hidden, some lethal, some merely discouraging. The thorns of a rose, the spines of a cactus, the stinging hairs of a nettle. These are the visible defenses, the ones we learn to avoid through painful experience. But the invisible defenses, the chemical defenses, are far more diverse, far more sophisticated, and far more deadly. Plants produce tens of thousands of different secondary metabolites, compounds that are not essential for basic metabolites, but serve other functions, primarily defense. These compounds include alkaloids, terpenoids, phenolics, and glucosinolates, and they can be toxic, repellent, or simply unpalatable, deterring herbivores, inhibiting pathogens, and shaping the interactions between plants and the animals that would eat them. Alkaloids are nitrogen-containing compounds that often have powerful effects on the nervous systems of animals. Nicotine, produced by tobacco plants, is an alkaloid that binds to receptors in the insect nervous system, causing paralysis and death. In higher concentrations, nicotine is also toxic to mammals, including humans, and tobacco plants produce it as a defense against herbivores. Caffeine, produced by coffee and tea plants, is another alkaloid, and it acts as a stimulant in humans, but in insects it disrupts feeding and reproduction. The plant is not producing caffeine for our benefit, it is producing it to protect its leaves and seeds from being eaten. Morphine, codeine, and other opiates produced by the opium poppy are alkaloids that have profound effects on the human nervous system, and they are used medicinally to relieve pain. But in the plant, they serve as defenses, deterring herbivores and perhaps also inhibiting the growth of competing plants. Terpenoids are the largest class of secondary metabolites, with more than 30,000 known compounds. They include the essential oils that give plants their characteristic scents, the menthol of mint, the limonene of citrus, the pinene of pine. These scents are not incidental, they are defenses, repelling some herbivores while attracting others, particularly predators and parasitoids of herbivores. Some terpenoids are toxic. Pyrethrin, produced by chrysanthemums, is a potent insecticide, disrupting the sodium channels in insect nerve cells, causing paralysis. It is used in many commercial insecticides because it is effective and it degrades quickly in the environment, but in the plant, it serves to protect the flowers from being eaten before they can be pollinated. Phenolics include tannins, lignins, and flavonoids, and they serve multiple functions. Tannins bind to proteins, making them difficult to digest, and they give plants an astringent taste that deters many herbivores. The bitterness of unripe fruit, the astringency of tea, the harshness of red wine are all due to tannins. Lignin is a structural polymer that makes plant cell walls rigid and difficult to break down, and it is one of the most abundant organic compounds on Earth, second only to cellulose. Lignin is not just structural, it is also defensive, making plant tissues tough and indigestible. Flavonoids include pigments that give flowers and fruits their colors, attracting pollinators and seed dispersers. But they also have antioxidant and antimicrobial properties, protecting the plant from oxidative stress and from infection. Glucosinolates are sulfur-containing compounds found primarily in the Brassicaceae family, which includes cabbage, broccoli, mustard, and horseradish. When the plant tissue is damaged, glucosinolates are broken down by enzymes to produce isothiocyanates, which are volatile, pungent, and toxic to many herbivores. The sharp taste of mustard, the pungency of horseradish, the bite of radish, are all due to isothiocyanates. These compounds deter most herbivores, but some insects have evolved the ability to tolerate them, and a few, like the cabbage-white butterfly, have even evolved to use glucosinolates as cues to find their host plants. This is an evolutionary arms race, with plants evolving new defenses and herbivores evolving new ways to overcome those defenses, a cycle that has been ongoing for hundreds of millions of years. Physical defenses are as important as chemical defenses. Thorns, spines, and prickles are modified stems, leaves, or epidermal outgrowths that make it difficult or painful for animals to feed on the plant. The thorns of a hawthorn, the spines of a cactus, the prickles of a blackberry, all serve to deter browsing by large herbivores. Some plants have trichomes, hair-like structures on the surface of leaves and stems that can be simple or branched, glandular or non-glandular. Glandular trichomes secrete sticky or toxic substances that trap or poison small insects. The trichomes of tomato plants secrete a sticky resin that traps aphids and other small herbivores. The trichomes of stinging nettles contain silica and histamine, and when they penetrate the skin, they inject these compounds, causing a painful burning sensation that can last for hours. Some plants use mimicry as a defense, resembling other plants that are toxic or unpalatable, thereby gaining protection without the metabolic cost of producing toxins themselves. This is Batesian mimicry, named after the naturalist Henry Walter Bates, who first described it in butterflies, but it also occurs in plants. Some non-toxic plants resemble toxic plants in leaf shape, color, or growth form, and herbivores, having learned to avoid the toxic model, also avoid the non-toxic mimic. Other plants use camouflage, blending into their surroundings to avoid detection by herbivores. Some alpine plants have leaves that are grey or brown, matching the colour of the rocks and soil, making them less visible to grazing animals. Some plants employ tolerance rather than resistance, allowing herbivores to feed on them, but compensating for the damage by growing faster or producing more leaves. This is a different strategy, one that accepts some level of herbivory as inevitable and focuses on minimizing the impact rather than preventing the damage. Grasses are particularly good at this. They grow from the base, from meristems located near the ground, so when the tips of the leaves are grazed, the plant can continue to grow, replacing the lost tissue. This is why lawns can be mowed repeatedly without killing the grass, and why grasslands can support large populations of grazing animals without being destroyed. Some plants have evolved induced defenses that are activated only in response to herbivore attack, rather than being constitutively present. This is metabolically efficient, because the plant does not waste resources producing defenses when they are not needed, but it requires the plant to detect the attack and to respond quickly. The detection is mediated by chemical signals, by the jasmonic acid and other hormones that are produced in response to damage, and the response includes the production of defensive proteins, the synthesis of toxic secondary metabolites, and the release of volatile compounds that attract predators of the herbivores. Induced defenses can be local, affecting only the damaged tissue, or systemic, spreading throughout the plant, providing protection against future attacks. The timing of defense is also important. Some plants produce higher levels of defensive compounds at certain times of day, timed to coincide with the activity patterns of herbivores. Some plants produce higher levels of defensive compounds in young leaves, which are more valuable to the plant and more vulnerable to damage, and lower levels in old leaves, which are less valuable and less vulnerable. Some plants produce higher levels of defensive compounds in reproductive tissues, in flowers and seeds, which are essential for reproduction and must be protected at all costs. The cost of defense is not trivial. Producing secondary metabolites, producing thorns and spines, producing trichomes, all require energy and resources that could otherwise be used for growth or reproduction. There is a trade-off between defense and growth, between defense and reproduction, and plants must balance these competing demands, allocating resources in ways that maximize fitness. In environments where herbivory is intense, high levels of defense are favored, even if they reduce growth. In environments where herbivory is low levels of defense are favored, and resources are allocated to growth and reproduction instead. Some plants have evolved mutualistic relationships with animals that defend them. Acacia trees in Africa produce hollow thorns and nectar-rich structures that attract ants, and the ants live in the thorns and feed on the nectar, and in return they attack any herbivore that tries to feed on the tree, biting and stinging the intruder until it leaves. The ants also clear away competing vegetation from around the base of the tree, reducing competition for light and nutrients. This is a mutualism, a partnership that benefits both the plant and the ants, and it is maintained by the plant's investment in housing and feeding the ants, and by the ants' investment in defending the plant. Other plants produce extrafloral nectaries, glands that secrete nectar outside of the flowers, on the leaves or the stems, and these nectaries attract ants, wasps, and other predatory insects, which feed on the nectar and also prey on herbivores that are feeding on the plant. The plant is bribing the predators, offering them food in exchange for protection, and the strategy is effective, reducing herbivory and increasing the plant's fitness. The diversity of plant defenses is staggering, a testament to the intensity of the selective pressure exerted by herbivores, and to the creativity of evolution, which has found countless solutions to the problem of being eaten. Every plant is a fortress, armed and defended, and every herbivore is a besieger, probing for weaknesses, evolving new ways to overcome the defenses. The interaction between plants and herbivores is one of the major drivers of biodiversity, shaping the evolution of both plants and animals, creating the complex web of interactions that characterizes terrestrial ecosystems. The defenses of plants are not just of academic interest, they have profound implications for human health and agriculture. Many of the medicines we use are derived from plant secondary metabolites: aspirin from willow bark, digitalis from foxglove, Taxol from yew trees. These compounds evolved as defenses, but we have co-opted them for our own purposes, using them to treat disease, to relieve pain, to save lives. And many of the challenges we face in agriculture are related to plant defenses. Crop plants have been bred for high yield and palatability, and in the process, many of their defenses have been reduced or eliminated, making them more vulnerable to pests and diseases. We compensate by applying pesticides, but this is costly and environmentally damaging, and pests evolve resistance to pesticides, just as they evolve resistance to plant defenses. Understanding plant defenses, understanding how they work, how they evolve, how they can be enhanced or manipulated, is essential for developing sustainable agriculture, for reducing our reliance on pesticides, for feeding a growing human population in a changing world. The green world is beautiful, but it is not benign. It is a world of chemical warfare, of physical barriers, of deception and mimicry, of alliances and betrayals, a world where survival depends on the ability to defend, to endure, to adapt. The plants we admire for their beauty, for their grace, for their tranquility, are also warriors, armed with toxins and thorns, engaged in a struggle for survival that has been ongoing for hundreds of millions of years, a struggle that continues today in every forest, every meadow, every garden, a struggle that shapes the world we live in ways profound and subtle, in ways we are only beginning to understand. The green world has survived 5 mass extinctions, ice ages that buried continents, droughts that lasted millennia, and the slow drift of continents across the face of the planet, and it endures still, carrying within its cells the memory of deep time. The genetic archive of 400 million years of adaptation. Plants are the most resilient organisms on Earth, not because they are invulnerable, but because they can wait, because they can endure conditions that would kill most animals, and because they reproduce in numbers vast enough to ensure that some individuals, somewhere, will survive even the most catastrophic events. The history of life on Earth is written in the fossil record, in the layers of sedimentary rock that preserve the remains of ancient organisms, and that record shows that plants have survived every major extinction event, every climate shift, every geological upheaval, and they have not merely survived, they have diversified, radiated, colonized new habitats, evolved new forms, new strategies, new partnerships. The green world is not fragile, it is ancient, tested, proven over timescales that dwarf human history, over timescales that approach the age of the Earth itself. The Permian Triassic extinction, the most severe extinction event in Earth's history, occurred approximately 250 million years ago, and it killed an estimated 90% of all species on Earth. The oceans became acidic and anoxic, the atmosphere filled with volcanic gases, temperatures soared, and the planet became for a time nearly uninhabitable. But plants survived. Spores and seeds buried in the soil waited out the catastrophe, and when conditions improved, they germinated. And the green world returned, slowly at first, then with increasing vigor, recolonizing the barren landscapes, stabilizing the soil, restoring the atmosphere, creating the conditions necessary for the recovery of animal life. The plants were the pioneers, the first to return, the foundation upon which the post-extinction world was built. The Cretaceous Palagine extinction, which occurred 66 million years ago and killed the non-avian dinosaurs, was triggered by the impact of a massive asteroid in what is now the Yucatan Peninsula. The impact threw billions of tons of dust and debris into the atmosphere, blocking sunlight, causing temperatures to plummet and halting photosynthesis for months or years. The forests burned, the food chains collapsed, and the dominant animals of the age perished. But again, plants survived, seeds in the soil, spores in the water, rhizomes and tubers underground, all waited in dormancy, and when the skies cleared and the sunlight returned, they sprouted, and the green world recovered. The extinction was selective. It favored plants with dormant stages, plants that could wait, plants that could survive without light for extended periods. The angiosperms, the flowering plants, which had evolved relatively recently and which had efficient seed dispersal and rapid growth rates, were particularly successful in the post-impact world, and they diversified explosively, becoming the dominant plants on Earth, a dominance they retain to this day. The ice ages of the Pleistocene, which began approximately 2.5 million years ago and ended only 10,000 years ago, saw massive ice sheets advance and retreat across the continents, covering much of North America, Europe, and Asia in ice kilometers thick. The climate was cold, dry, and unstable, with rapid shifts between glacial and interglacial periods. Many plant species went extinct, unable to migrate fast enough to keep pace with the shifting climate, but many others survived, retreating to refugia, small pockets of suitable habitat where conditions remained tolerable, and from these refugia they recolonized the landscape when the ice retreated. The genetic diversity of modern plant populations reflects this history of contraction and expansion, of bottlenecks and founder effects, of survival in isolated refugia and subsequent dispersal. The plants that survived the ice ages were those that could tolerate cold, that could disperse their seeds over long distances, that could colonize disturbed habitats quickly. And these traits are still evident in the plants of temperate and boreal regions today. Climate change is not new to plants. They have experienced climate change throughout their evolutionary history, and they have responded by migrating, by adapting, by evolving. But the current episode of climate change, driven by human activity, by the burning of fossil fuels and the release of greenhouse gases, is occurring at a rate unprecedented in recent geological history, and it poses challenges that are different in kind, not just in degree. The climate is warming faster than plants can migrate, habitats are fragmenting, barriers to dispersal are increasing, and the composition of the atmosphere is changing in ways that affect photosynthesis, water use, and nutrient cycling. Some plants will adapt, some will migrate, and some will go extinct, and the green world of the future will be different from the green world of today, shaped by the pressures of a rapidly changing climate and by the actions or inactions of humanity. Plants are already responding to climate change. Spring is arriving earlier and fall is arriving later, extending the growing season in many temperate regions. Plants are leafing out earlier, flowering earlier, and fruiting earlier, tracking the warming temperatures. Some species are shifting their ranges poleward or upward in elevation, following the climate zones to which they are adapted. But not all species can migrate. Plants with limited dispersal, plants that depend on specific pollinators or specific soil types, plants that are confined to isolated habitats like mountaintops or islands may be unable to move, and they may face extinction as their current habitats become unsuitable. The rate of climate change is the critical factor. If the climate changes slowly, plants can adapt through natural selection, evolving new traits that allow them to tolerate the new conditions. But if the climate changes rapidly, adaptation may not be fast enough, and extinction becomes more likely. The resilience of plants lies in their diversity. There are approximately 400,000 species of plants on Earth, and they occupy nearly every terrestrial habitat, from the wettest rainforests to the driest deserts, from the equator to the poles, from sea level to the highest mountains. This diversity is not just taxonomic, it is also functional, ecological, genetic. Different species have different tolerances, different strategies, different partnerships, and this diversity ensures that no matter what happens, some plants will survive, some plants will thrive, and the green world will continue. Biodiversity is resilience, and the loss of biodiversity, the extinction of species, reduces resilience, makes ecosystems more vulnerable to disturbance, more likely to collapse under stress. Conservation of plant diversity is essential, not just for the sake of the plants themselves, but for the sake of all life on Earth, including humanity. Plants provide the oxygen we breathe, the food we eat, the materials we use to build our homes and make our clothes, the medicines we use to treat disease. They regulate the climate, stabilize the soil, purify the water, and create the habitats that support the diversity of animal life. The loss of plant diversity is a loss of ecosystem services, a loss of resilience, a loss of options for the future. Protecting plant diversity means protecting habitats, reducing pollution, mitigating climate change, and managing ecosystems sustainably. And it also means preserving seeds, maintaining botanical gardens and arboreta, and supporting research into plant biology, ecology, and evolution. The seed banks, the repositories of genetic diversity, are a form of insurance, a hedge against catastrophe. The Svalbard Global Seed Vault, buried in the permafrost of a Norwegian island, contains more than 1 million seed samples from around the world, representing thousands of species and varieties, a backup of global agriculture, and a repository of wild plant diversity. If a crop variety goes extinct in the field, if a wild population is lost to habitat destruction or climate change, the seeds in the vault can be used to restore it, to reintroduce it, to preserve the genetic diversity that may be essential for future adaptation. This is not a solution to the biodiversity crisis, but it is a safeguard, a way of buying time, of ensuring that the genetic resources of the green world are not lost forever. Plants also hold the memory of the past in their DNA, in the sequences of genes that have been passed down from generation to generation, accumulating mutations, recombining, evolving, but also preserving information about ancient adaptations, ancient partnerships, ancient environments. By studying plant genomes, by comparing the DNA of different species, we can reconstruct the evolutionary history of plants. We can identify the genes that are responsible for specific traits, and we can understand how plants have adapted to different environments, how they have responded to past climate changes, and how they might respond to future changes. This knowledge is essential for predicting the impacts of climate change, for developing strategies to conserve plant diversity, and for breeding crops that are more resilient, more productive, more sustainable. The future of the green world is uncertain, but it is not hopeless. Plants have survived worse than what we are currently facing, and they will survive this too, though the composition of the green world may change, and some species will be lost. The question is not whether plants will survive, but which plants will survive, and what the world will look like after the current wave of extinctions has passed. The answer to that question depends in large part on the choices we make, on the actions we take, on our willingness to protect habitats, to reduce emissions, to value biodiversity, to recognize that the green world is not a resource to be exploited, but a living system of which we are a part, a system that sustains us, that makes our existence possible, and that deserves our respect, our care, and our protection. The green world is a testament to the power of life to endure, to adapt, to persist through unimaginable challenges, through extinctions and ice ages and asteroid impacts, through the slow grinding of geological time. It is a reminder that life is resilient, that it finds a way, that it endures, and it is an invitation to us to join in that endurance, to become partners in the survival of the green world, to recognize that our fate and the fate of the plants are intertwined, that we depend on them as much as they in some ways have come to depend on us, through the landscapes we shape, the climates we alter, the ecosystems we manage or mismanage. The green world has been here for 400 million years, and it will be here long after we are gone. But what it looks like, how diverse it is, how resilient it is, depends on what we do now in this moment. In this brief window of time when we have the power to shape the future, for better or for worse. The silence of the green world is not the silence of absence. It is the silence of patience, of endurance, of deep time. It is the silence of roots growing in the darkness, of seeds waiting in the soil, of leaves turning toward the light, of the slow, steady accumulation of carbon, of oxygen, of life. It is a silence that speaks, if we are willing to listen, of resilience, of adaptation, of the long memory of evolution, of the partnership between life and earth that has shaped this planet and made it habitable, made it beautiful, made it home. And in that silence, in that patience, in that endurance, there is hope, not the shallow hope of easy solutions, but the deep hope of life itself, the hope that has carried the green world through 400 million years of change, and that will carry it forward into the future, into the unknown, into the long, slow unfolding of time.